welcome to my first ever YouTube video. Welcome to IB Biology Absorption. The reason I'm doing this channel and making these videos is to earn CAS hours. If you're an IB student like I am, you know what CAS is, and you know that you need CAS hours. So I'll be exploring and explaining the syllabus from my point of view, combining what my teachers have taught me, plus the materials that we're given. And just to put it out there, I am in no way whatsoever an expert. I'm just a high school student who is trying to master all these concepts that we have to learn for IB Biology. And another disclaimer is that I'm very new to making videos and recording and teaching as well. So if you have any tips or feedback, anything at all, please comment in the comment section and please leave a thumbs up if you enjoyed the video, and please subscribe if you would like to see more content. So yeah, without further ado, today we'll be covering 1.1 Introduction to Cells from Topic 1, Cell Biology. So the cell theory as we know of it today was not created on a whim. It was actually developed by many scientists throughout the years, and it took a lot of time for it to get where it is today. So we can summarize the cell theory in 3 statements. Number 1, living organisms are made up of one or more cells. So one cell meaning multicellular, more cells meaning, one cell meaning unicellular, and more cells meaning multicellular. And the second rule is that there is no smaller unit of life than cell. So nothing can exist and function on its own as a form of life other than a cell. And the last one is pretty obvious. Every new cell must come from a pre-existing cell. So cells do not just spontaneously appear. And yeah. So 3 scientists play a key role in the cell theory, and they are Schwann, Schleiden, and Virchow. I will link a video explaining who these scientists are and what their contributions were to developing the cell theory in the description box, so please check that video out. It's very interesting. It's from TED Ed, and yeah. So of course there are still some exceptions to the cell theory, but this does not mean that we can just throw out the theory out the window. We just have to keep these theories in mind because they're very rare and they're not frequent at all. So the first cell that IB would like you to know is from the Acetabularia species called giant algae, and this is what it looks like. So it's quite big, and it can be seen with the naked eye, and it challenges the idea that single-celled organisms, so unicellular organisms, they should be small and simple in structure, shape, and size. But this is not the case for this specific cell. It can be held in your hand. It's quite large, and it can be up to 10 centimeters in length, and it has just one single nucleus. The next one is the striated muscle cells. This muscle cell is striated, meaning it's like striped and separated. So it can be very long, up to 30 centimeters in length, and they have multiple nuclei. So as you can see, this section right here is one whole cell, and it has a lot of nucleus. So 1, 2, 3, yeah, more than 3 nucleuses for sure. And this challenges the idea that a cell should have one single nucleus that operates the entire cell. And the last one that IB wants you to know, the last one is called aseptate fungal hyphae, and this is what it looks like. It is basically a kind of fungi, and it doesn't have any dividing walls. So it's aseptate, septa meaning wall, separating, a meaning none. So there is no wall here, unlike in this one where it does have a wall. And this challenges the idea that a living structure should have specific, discrete, singular compartments rather than be one single flowing organism. I won't go too much into detail for this one, but just keep in mind that you have to know very, very well how to convert from millimeter to micrometer to nanometer. It is very, very important because IB will definitely give you one of these questions where they will present you with an image of a cell and they will ask you to calculate its magnification or the actual size or the size of the drawing or whatever. So this diagram, if you can kind of remember, it's very handy to remember. And yeah. So all living organisms have to display these 7 functions of life for them to be considered living. First one, metabolism. Metabolism is basically the process where all the material that is consumed by the cell is created into or transferred into energy. That is what metabolism is. So some of all the reactions happening within the organism or within the cell, and this is what all living organisms have to have the ability to do. So they have to have the ability to take in food and convert it into energy. Reproduction, production of offspring, so they should be able to create offspring either sexually or asexually. Sensitivity, so they should be able to respond to touch, to any kind of obstruction, both internal and external. Homeostasis, they should be able to maintain a stable internal environment. So if a plant cell has too much water, the vacuole should be able to notice that and get rid of the water somehow, so that every other reaction or every other thing that's happening within the cell can still continue. Excretion, so removal of waste products. So after metabolism, all the waste products should be removed, should be excreted. Nutrition, so intaking materials, consumption of materials. So for us, eating food. Growth, meaning movement and also change in shape and size. So the IB would like you to know 2 unicellular organisms, but I heard it varies from school to school, so you should check in with your biology teacher on which unicellular organisms you guys will study. So for us, it was Paramecium and Chlamydomonas. We'll start with Paramecium. So reactions are catalyzed by enzymes in the cytoplasm in a Paramecium cell. So this is all the cytoplasm, and this is where metabolism takes place. Reproduction can be asexual or sexual. Homeostasis, the contractile vacuoles, which are these sunflowery things, they remove excess water, and this is how they maintain a stable internal environment. Nutrition, they take in digested organic materials. They absorb digested organic materials, and they have specific vacuoles for these. So these food vacuoles are where all these materials that they take in, the smaller organisms, are digested. Paramecium are heterotrophic. For excretion, CO2 diffuses out of the cell for cellular respiration. Stimuli, when facing an obstruction, the paramecium uses its cilia to move around. Growth, it grows by the absorption of digested organic materials. And yeah. Chlamydomonas, similarly, has its reactions catalyzed by enzymes within the cytoplasm. Also, reproduction is asexual or sexual. Maintains an stable internal environment by removing excess water with the help of their contractile vacuoles. These are the contractile vacuoles right here. Nutrition, it is a photosynthetic organism, so it gets its nutrition from photosynthesis. Excretion, it excretes O2 during photosynthesis and CO2 during cellular respiration. Both these gases diffuse out of the cell. Stimuli, the eye spots can sense brighter light, and using its flagellum, it moves towards the light. So similarly, if it's in a colder environment, it will use its flagellum to propel towards a hotter environment. And yeah, growth, it grows through the uptake of inorganic mineral ions and of course through photosynthesis. The rate of metabolism of a cell is dependent on the mass or volume. So you can imagine that a larger cell needs more energy, right? So if a cell has a larger volume, it means that the metabolic rate would be really, really big and fast. But a larger surface area would mean that the cell would have a greater rate of material exchange. So how can these two traits be combined so that a cell can do its best performance? So you can imagine it as a cube. The cube does have doesn't have a lot of surface area, but it does have a lot of volume. So what a cell does is it maintains its volume, but the surface area increases. And this is why cells should remain small when they divide. Yeah, so ideally cells should divide and remain small so that they can maintain a high surface area to volume ratio. So the statement that Aristotle gave us, the whole is greater than the sum of the individual parts. What does this mean? It means that multicellular organisms, and not unicellular, multicellular organisms that are comprised of multiple different types of cells show emergent properties, meaning that all these cells work together to produce a system. And you can imagine this for us humans or animals. Cells become tissues, tissues become organisms, organs form a system, and systems can collectively carry out all the functions that are necessary for us to live. And thank you, Aristotle, for this great, great statement. Differentiation is a process that happens during the early stages of development. So when new cells start to become specialized and different from other cells, some genes tend to be expressed and some genes are not. So us multicellular organisms, we have cells that have their own specific functions. A skin cell can't suddenly do the job of an eye cell, and vice versa. So during cell division, cells make sure that every single cell is identical and they have all the genes that are necessary for that cell to function. And cellular differentiation is where some of these genes that cell has are expressed, some are not. And my teacher describes it as being in a house where some lights are on, some lights are off. You might leave the light in the living room on because you want to be in the living room, but you might keep the light in the bathroom off because you don't want to be there. And yeah, each cell has a specific gene or subset of genes that they express, which determines what they will be doing. So through gene expression, through the subset of genes that become expressed, it codes for proteins that the cell will create, which will determine the function of the cell. Here you can see a stem cell is the earliest form of a cell, and it has the ability to become any kind of specialized cell. So when a stem cell becomes a muscle cell or fat cell, it loses this ability to become any other cell. So stem cells are cells that have the ability to develop into many different types of cells in the body. So this begins during fertilization, where the egg cell is fertilized and a zygote is formed. And during this formation, the first cells are known as totipotent stem cells. Totipotent stem cells can differentiate into any cell type and even give rise to a completely new organism. Stem cells have their own, you can imagine it as a hierarchy. So this is the top hierarchy that gives the highest level of potency. The highest level of potency meaning that it has the largest ability to differentiate. The next level would be pluripotent stem cells, which doesn't have as much of an ability to differentiate as totipotent stem cells do. Pluripotent stem cells can differentiate into any kind of body cell, and they are the embryonic stem cells of blastocytes. The next level is multipotent stem cells, and these can differentiate into closely related cells, and these cells are from the umbilical cord, and they're usually known as umbilical cord stem cells. And this is the last level, unipotent stem cells. These cells can only differentiate into their specific cell type, and they are usually called adult stem cells. For example, you can see when the when the egg cell is fertilized, it becomes a morula, and the first few cells within the morula that make up the morula are totipotent. These cells can give rise to any kind of cell type. Then the morula gets bigger and it becomes a blastocyte, which becomes a human fetus, and then the cells of the blastocyte are pluripotent, and it goes on and on. So from totipotent to pluripotent to multipotent to unipotent. Stem cells are widely used to treat various diseases, and these are called stem cell-based therapies. Different cells can be grown, such as skin cells using skin stem cells, Embryonic stem cells can also differentiate into any kind of specialized cell. However, we have to keep in mind that using embryos does have certain ethical implications, but we'll talk about those later. The most widely used ones, after lots of research of course, are umbilical cord blood stem cells and adult stem cells, but of course they are limited in differentiation. So the first disease that uses the help of stem cells for its treatment is Stargardt's disease. It's a genetic disease that causes degenerate macular vision loss, and it leads to a progressive loss of vision. So stem cells from days-old human embryos differentiate into retina cells, which are then injected into the patient's eye. And so far, no harmful effects of this treatment have been found. And the next one, depending on what your school teaches you, we learned about leukemia. So you'll need to check in with your IB biology teacher on what other stem cell related disease you will need to learn. So for leukemia, it's a cancer that causes a high number of abnormal white blood cells that are produced by the patient's bone marrow. Adult stem cells, which are taken from another bone marrow, are injected into the patient, and these blood stem cells enter the bone marrow via the bloodstream, and the patient's already existing cancer cells in their bone marrow are eliminated through chemotherapy. Now, there are many discussions and debates regarding the ethical issues of using stem cells. Adult stem cells are fine for use as long as consent is taken from the patient, from the donor. Cord blood stem cells are also fine to use as long as requirement and consent is taken from the parents of the child. However, stem cells from embryos created in vitro, basically mean embryos that are created specifically for couples that are sterile, so they don't have the ability to have children. So creating embryos just for the purpose of being able to treat and give rise to different cell types has been a long-standing debate, depending on where your beliefs lie on where human life begins. So there's a lot of ethical, messy issues behind this. So yeah. Thank you for watching. I hope it was okay. I hope you kind of learned something, I guess. I will leave my study guide for chapter 1.1 in the description box, so please check that out if it helps you study. Send me an email if you have any questions, or of course use the comments. Yeah, thank you. Thanks.